Welcome everyone to the 2021 New Limbs of Life Symposium. Today is the last session of our seminar series, and you will hear about latest methods that may help us to discover the currently undiscovered. Thanks again for joining. As in previous sessions, we will have two main speakers today, followed by a brief Q&A. We also have 4 short talks that we selected from the abstracts. And after the official webinar is over, please don't forget to join us to the breakout session, which you can access through a separate link that is to a regular Zoom. There you will then have the opportunity to directly ask questions to the presenters to discuss their work in more detail. Now, I'm really delighted to announce the moderator of today's session, Dr. Tanja Woike. She's a deputy director at the Joint Genome Institute, where she's leading the user program, the microbial program, and the single cell group. Dr. Woike, please take it from here.
Thank you, Frederick, for the great introduction. It's a really great pleasure to be the moderator today. I'm very excited. And it's a even better pleasure to introduce our first speaker today, which is Alicia Clum from our Joint Genome Institute at Lawrence Berkeley National Laboratory. Alicia has been at the JGI since 2006, when she started during the Sanger days and with a focus on the wet lab genomics. In fact, she helped us at the time close gaps in single cell genome sequence data. As next generation sequencing came along, she transitioned into data analysis and bioinformatics. And more than 7 years ago, she took over as the lead of the genome assembly and analysis group. Alicia has a very broad, but at the same time, very deep knowledge in sequence data processing. And with the ever-increasing growth of sequence data, she's become quite interested in tackling challenges that come along with processing this data efficiently. And with that, she's gonna tell us today about a really neat project that involves dealing with this big data in a talk that's entitled MetaHipMer for Terabyte Scale Assemblies. Take it away, Alicia.
Thanks for the introduction, Tanja. Can you see the slides?
Yes, looks
okay.
good.
So thanks everyone for coming to listen to my talk today. I'll be talking about MetaHipMer for Terabyte Scale Assembly. So I'll briefly give an overview of the JGI for those of you who aren't familiar, and then I'll move on to background on metagenome sequencing and why we care about computational resources for assembly. Then I'll talk about MetaHipMer and how we can use supercomputers to generate these really large data sets. And finally, I'll give an example of supporting a JGI user project with these tools. So the Joint Genome Institute is part of Lawrence Berkeley Labs. We're funded by the Department of Energy, and our mission is to provide the global research community with genome science capabilities. We are focused on mainly a couple of important DOE mission areas, such as bioenergy, carbon cycling, and biogeochemistry. We do this through a number of scientific programs and capabilities, and today I'll be focusing on how we're supporting metagenomics users using sequencing and computational analysis. So this is a figure from our JGI strategic plan showing the growth in both genomes and metagenomes over time, with the orange box here showing how we anticipate this to continue to grow. So we're expecting a growth in data for both genomes and metagenomes, and this creates a bit of a problem, and we're trying to figure out how to deal with it. So MetaHipMer is a tool that we can use to try and tackle that problem. So not only are we generating more data sets, but those data sets are also larger. So this is a historical view of sequencing platforms and their cost and throughput over time. It's a bit out of date, but gives you the general idea that over time, in the black line, the throughput of sequencers have dramatically increased, and in the orange line, the cost has dramatically decreased, famously faster than Moore's law. And so kind of where we're at today is we're using the Illumina NovaSeq with S4 flow cells, which can generate up to 2.4 terabase pairs per run at a cost of about $6 per gigabase pair. And when we brought this machine on in 2017, one of the things that we did with the extra capacity was to provide more data per project for metagenome drafts. The other thing that's driving this throughput is an increase in lab automation. So for example, users used to maybe send in a handful of samples, and now they're sending in plates worth of samples for us to process. And so between this increase in the number of projects and the increase in the number of data per projects, we really need some new tools to deal with the data. This is a general overview of the JGI workflow. So we have users around the world that collect samples and extract DNA. That DNA is shipped to the JGI, where we make libraries and generate sequencing data. Most of the metagenomes use short read technology, and then that data is assembled to try and reconstruct the microbial community. We then do annotation and try and generate metagenome assembled genomes. So here we're trying to reconstruct one pile of contigs per organism in the community, and these end up being having a variety of completeness and contamination. So it's important for context to understand that metagenome samples vary in complexity. Hopefully everyone's pretty familiar with this idea. So here we've plotted a couple of common habitats from least to most diverse on the x-axis, along with the sequencing diversity score from a tool called NonPreal3 on the y-axis. So it's important to understand that samples vary by complexity because the resources required to assemble these different habitats is a function of the complexity. And that's true because most short read assemblers use something called de Bruijn graph algorithms to process the data. So with de Bruijn graphs, reads are broken down into overlapping substrings or k-mers for efficient memory storage. So in our little illustration here on the right, we have a sequencing read in yellow, and then we've broken down in the constituent k-mers. Here, the first and the last k-mer, ACGA, are the same, excuse me. And so we only have to store this once. So this is a bit more memory efficient. Most of these algorithms, popular tools like Metaspades and MegaHit, are designed to run on just a single computer node, and so they're limited by how much memory is on one machine. So the more unique k-mers that you have, the more memory that you need. So the amount of unique k-mers is a function of both sample complexity, which we just talked about, and sequencing depth, which we'll talk about shortly. So here I have a figure showing for Metaspades, which is a popular tool in the community. On the x-axis, the number of unique k-mers in billions versus the memory in gigabytes required to assemble the data. So as the number of unique k-mers increases, so does the computational resources required to process the data. So if we go back to our habitats, something like a bioreactor is going to be lower resources than something like a freshwater lake to assemble, and soil is going to be one of the most resource intensive habitats to deal with. What we found is that many habitat types are being under-sequenced. So at the JGI, a lot of PIs prefer to send more samples and sequence them at a more shallow depth than to sequence a smaller number of samples at a larger depth. So we wanted to understand just kind of how far off we were. So again, here we're using NonPreal3 with these same 5 habitats plotted from lowest complexity to highest complexity on the x-axis. And here we're looking in blue, how much data did we generate versus estimates in red of from NonPreal3 about how much data would be required to saturate the library to 95%. And then on the second x-axis, I've plotted in these black dots the median sequence diversity from a couple slides ago. So what you see here is that for pretty much everything but an engineered habitat, we're under sequencing the data. And the habitat kind of determines just how far we're off. So for freshwater, we're off by, maybe an order of magnitude, and for soil and sediment, we're off by, maybe 2 orders of magnitude. And so what this means in practice is that we end up with low coverage, which results in incomplete and fragmented assemblies. One way to try to address this is to combine data that makes sense biologically. So there's some different examples of how we could do this. If we had data from different seasons, we could assemble each season together. If we had data from different geographic locations, for example, you could assemble everything from the Black Sea together. And if we had soil cores, we could assemble everything from a similar depth range together. Combined assemblies are not new. We've been supporting this at a low level at JGI for quite a while, but because of the computational resources, we've been limited either in terms of the number of samples we can combine together or how many of these we can do total. And so something that we've been working on for the last couple of years is a tool called MetaHipMer, which is a distributed assembler. So MetaHipMer is part of a suite of bioinformatics tools that are funded by Exabiome, which is funded by DOE through this exascale computing project. So what is exascale? Exascale computing is a system that's capable of 1 billion or 10 to the 18th calculations per second. So it's an exaflop. So it's based on how many calculations it can do per second, not how much memory the machine has. And so this tool is really developed to run on really large supercomputers. So if we look at this illustration on the right, the difference between a shared memory algorithm like Spades or MegaHit is that you're limited by the memory on just a single node. So if this node had 100 gigabytes of RAM, that would be your maximum memory. With a distributed approach, you can access memory across multiple nodes. So here in this illustration, we could potentially access up to 400 gigabytes if each of these nodes had 100 gigabytes. And so this can be a really challenging problem as the communication between nodes is difficult. But if you can get this to work and get it to scale, you can really have a much larger amount of aggregate memory. So we've been working mostly on 2 different supercomputers. The first is Cori, which is at NERSC at Lawrence Berkeley National Lab. And this is technically not an exascale system. It's a petabyte scale or petascale system. And it has 1 petabyte of total memory. And then for the really large runs, we've been using Summit, which is at Oak Ridge National Lab. And that is technically an exascale system. It was the fastest supercomputer in the whole world when it came out. And I believe currently it's the 2nd fastest system. And it has over 10 petabytes of total memory. So just how much data can we assemble with MetaHipMer? Here I've plotted the largest assembly or the largest input data size that we've been able to process per year. So in 2019, we were able to assemble about 3.3 terabytes. In 2020, we spent a lot of time on scaling and we're able to bump this up to almost 8 terabytes. And then in 2021, we were able to assemble up to 25 terabytes. I'll use both bytes and bases in this talk. I tried to distinguish them by using BP to specify base pairs. And there's 2 bytes per base. So if you want to convert into bases, you would just divide these numbers by 2. The project overall has a goal of supporting up to 50 terabytes of input data by 2023. So now if we overlay the memory that was requested to generate these assemblies, we're using anywhere between 100 to almost 1,000 terabytes of RAM. So this is just not something that people have access to on a single node. So for example, most of the Cori nodes, if you're just looking at one node, either have 64 or 128 gigabytes of RAM. So if you can really only process these really large data sets if you're using a large system and you're using a large amount of total aggregate memory. So these assemblies used a couple hundred to on the order of 1,000 nodes. So now I'll walk through a couple of the figures from the MetaHipMer paper, which was published in 2020. So I just want to walk through the experimental setup, the different approaches, and then a couple of the figures. So some of the figures from the paper use this WA mix data set. So they took, they used a tool called CamiSim to generate 12 synthetic data sets containing 25 references from MARREF, which is a curated marine reference genome database. And then they mixed those with 12 real samples from Western Arctic Ocean samples, which the total size of those between the 12 samples was 822 gigabytes total. So we've really generated a new set of data with 12 samples each, and each of those samples is a mixture of real data from a Western Arctic Ocean sample and one synthetic spike in. And then to evaluate the assembly results, we're using MetaQuast with the 25 MAR references as the reference database. And so here are some cartoons of just the 4 different approaches that are shown in the figures. So the 2 main approaches here are co-assembly or multi-assembly. So co-assembly is where you take reads from different samples that make sense biologically to combine together, and you put them all into the assembler and you generate a set of contigs. Multi-assembly refers to what most people are doing, where each sample is assembled individually. So sample 1 gets its own set of contigs, sample 2 gets its own set of contigs, and then you're left with trying to combine all of the results. Here in green, we're showing it's the same as the multi-assembly, but there's one additional step where you're trying to dereplicate redundant information. In practice, a lot of people wait until the mag level to do this, but for the publication, they're attempting to dereplicate the contigs. And then just for comparison, you wouldn't do this in practice for metagenomes because we don't have references, is also displayed is assembling the samples individually and then picking the best, the most complete for each organism from within all of the contigs. So organism 1, the best, most complete result for organism 1 might be from sample 2. For the second organism, it might be from sample 1. And so in the plot, we've kind of cherry picked the best results per organism for the 25 references. These next 2 figures are only showing the synthetic data. So you do the assembly of the data mixed together, and then we've kind of fished out just the results, the assembled contigs from the spike-ins to assess what's going on. This is a cumulative length plot from MetaQuast, which is a useful tool for evaluating assemblies with or without a reference. Here we have a reference, so that's enabled, and so that's represented in the gray dotted line right here. And so we're plotting the number of contigs on the x-axis versus the cumulative length on the y-axis, and the contigs are ordered largest to smallest. And so what this means is that the steeper the slope of the line and the more quickly it reaches this reference line, the better. So that means you have longer contigs and therefore fewer of them. So what you're seeing here is the co-assembly in blue has the best contiguity and reaches the total expected genome size the fastest. So this is the best assembly. What you see in orange and green are the multi-assembly and the deduplicated assembly. You'll see that both of them are over this gray line, and that is indicative of redundant content. So you have a high duplication rate And even after attempting to deduplicate, it doesn't work fully. And so you're left with some duplicate content. And then in red here, this is the worst approach because it's got the flattest slope and is also incomplete. Here we're plotting the genome fraction recovered as measured by MetaQuast versus the simulated sequencing depth. So we're using the same colors, same labels here, and what is plotted on the x-axis is the sequencing depth of the simulated data for an individual sample. So if we intersect this graph vertically, for example, you'll get the same data from all the same organism. So here, these arrows point to an organism where actually the combined assembly did worse because there were two closely related strains, and that's something where MetaHipMer struggles a little bit. That's not unique to MetaHipMer. It's something that all assemblers may struggle with if there's closely related species or strains. So what you can see here is when there's organisms at low abundance, say less than 20x, the genome fraction is really low. And it's only when you combine the data to then get a higher observed depth than what you could get from an individual sample, you can assemble some of these data sets at much higher completeness, most of them being above 90%, because you're effectively, in this case, multiplying the coverage by 12. For a real data set, this wouldn't necessarily be true. It wouldn't necessarily be this good because the abundance is going to vary by sample. Whereas in this simulation, all 12 of the spiked in data sets that were generated had the same coverage for the same organism, but the reads just have different start and stop locations. So for some of the other approaches, the multi-assembly and the deduplication, you really don't start to see recovery above 90% until about 60x. And for an individual sample, you really don't start to see it until, over 100x. This is kind of a function. This is a function of how De Bruijn graph assemblers work. And so if you were using a different approach, for example, long read high accuracy PacBio data, the amount of coverage that you need is much lower, say about 30x. So we've talked mostly about reference-based analysis of these individual genomes, but what do you do if you don't have a reference? So in 2017, there was a community-led effort to define standards for these metagenome assembled genomes, or MAGs. So this was led by Robert Bowers at the JGI, and it sets 3 criteria. So it sets an assembly quality criteria, a completeness criteria, and a contamination criteria. So for example, to be a medium quality draft MAG, you need a completeness of greater than 50% and a contamination of lower than 10%. So currently, high quality and medium quality MAGs are being stored and labeled in IMG. And so if we go back to our figure that we were just looking at, and we draw a line at 50%, just one dimension of defining MAG quality, what you see is that 24 out of 25 of the co-assembled organisms would end up being medium quality, at least in the dimension of completeness. Whereas all of the other approaches, you would end up with fewer MAGs being generated. So in the case of best sample, and here we're cherry picking the best, you only end up with 5 MAGs. So you get 5 times more with a combined assembly approach. So this is definitely one reason to try co-assembly. So you'd end up with more MAGs. All right, so simulated data is great, but let's talk some about real data. So For the last couple of years, we've been supporting a number of JGI user proposals. We've generated MetaHipMer assemblies for 10 user proposals, and we'll be taking a deeper dive into the Great Redox experiment in wet tropical soils, which is our second largest data set to date. So this data is from Jennifer Petridge's lab. She's the one that generated the samples. They're doing sampling from this tropical forest in Puerto Rico, here in green, and here's a couple photos from their sampling trip. So what they were really after was to understand how changing redox periodicity impacts community structure and function and carbon flux. So in this diagram, we have an oxic environment, which means there's free molecular oxygen, the anoxic environment on the bottom, which means there's no free molecular oxygen, and then two conditions where you're fluctuating between oxic and anoxic at different frequencies. And so they're using different conditions, time points, as well as stable isotope probing, which uses substrate labeled carbon-13 to try and answer their questions. And so we ended up generating, they sent 96 samples. We generated 22.6 billion reads. And so we'll focus just on an attempt to combine the entire data set together. So they had on their own generated some combined assemblies with smaller subsets of the data, but they weren't able, because of the size of the data set, to do a single combined assembly. So here's another cumulative length plot from Quast. Here we don't have a gray line, horizontal line, because we don't have a reference. So here we're plotting the cumulative length by the number of samples in a data set. So you can see that with one sample, the assembly is quite small. And as you increase the number of samples, both your total assembled size increases as well as your contiguity is improving because the slope of this line is getting steeper. So by the time we assemble all 96 samples, which is 7.7 terabytes of data, you end up with 775 gigabases of assembled data, with 87% of the data mapping back, which is pretty high, especially for a soil sample. So that means about 20 million reads mapped back to the assembly. So here we're really demonstrating that co-assembly can lead to a total larger assembled size and increased contiguity. So now if we look at those same assemblies, we can generate mags using MetaBat2 and look at how many medium and high quality bins we can get. So here we've, on the x-axis is the number of subsets, which have also been labeled with the data set size. And then the bars are the number of mags we generated. So there's blue quality mags. Sorry, there's medium quality mags in blue, and in red we have the high quality mags. And we've overlaid a couple of lines just to give you some context. So the turquoise line is the number of mags we typically see from a soil sample, and the yellow line is the number of mags that we typically see from all of the habitats combined. And so what you can see is that even combining a couple of these samples, you're already getting more mags than you would see for an individual or this multi-assembly approach. And then when you get all the way up to 7.7 terabytes, you're getting over 100 mags. And so you're getting an order of magnitude more mags than we'd recover from a typical genome. For an apples to apples comparison, we treated all the reads here as if they were from a single sample. A lot of binning tools, if you generate the depth information by sample, you can recover additional bins. And when we do that for the 7.7 terabyte data set, we end up doubling the number of bins by another factor of 2. So Robert Riley is going to give a flash talk on taxonomic analysis of the co-assembly bins. So I hope everyone has a chance to join for that talk. So in conclusion, assemblies of terabytes of sequencing data is possible using supercomputers and MetaHipMer. We have an increased likelihood of novel discoveries compared to traditional approaches because you're generating larger and more contiguous assemblies. You can generate more total mags, and you're going to have a higher depth of genomes occurring in low abundance in individual samples. Some of the other benefits are a unified set of contigs and bins without needing to dereplicate, which can be a messy process, and a simpler workflow. And then just a note of caution, if you're expecting high strain heterogeneity, some of that might get collapsed. Acknowledgements. A big thanks to Stephen Hoffmeyer and Rob Egan, who've been doing a lot of the development work, and Robert Riley from the JGI has been leading the efforts internally to generate these user assemblies. There's a lot of other people at the JGI that are involved in this project or have contributed to the data in this deck. Ronan O'Malley and his team generated all the sequencing and libraries in the lab. Jennifer Petridge and Trina McMahon's lab have been very patient in letting us use their data to work through some of these scaling problems. The exobiome work is funded by the Exascale Computing Project. The assemblies would not have been possible without NERSC and Oak Ridge to run on. And then the JGI is primarily funded by Department of Energy Office of Science. I've included a couple references just for if anyone wants to look into anything in more detail, or if you'd like to contact us. And if there's time, I'll stop sharing and take some questions.
Great talk. Thanks, Alicia. So we have time for some questions. I don't see any yet, but maybe I'll get started with a question. You mentioned for the Petridge, the GRE Great Redox experiment, that 87% of the reads mapped back to the core assembly. These remaining 13%, what do you think? Is it eukaryotes? Is it really low covered organisms? Or have you looked at that non-map data? What is it?
We have not looked at that at all. Okay. It's probably both.
Yeah. Okay. Let's see. Still no questions. I can keep going. You mentioned you showed the data, your benchmark data showed the multi-assembly and the dedupe, and it showed that it kind of exceeded the reference. So there was redundancy. Why is that happening? What's going on there?
What's happening is that if you're assembling the samples individually, and if you have the same organism at high depth, those are going to be assembled in both data sets. And dereplication is just a messy process, either at the contig level or at the bin level. So way back in the day, we used to assemble different k-mers and then combine them. And we were delighted when we didn't have to do that. So anywhere where you can just eliminate that step completely, you're going to be better off.
Okay, great. And we did get a question in the chat here from George Schaible. How easy is it for researchers to access supercomputers to use programs like MetaHipMer?
Not that easy. So if your work is related to Department of Energy, you can submit applications to get time on these systems. So Corey specifically has a goal of supporting all of the offices of science. They just actually had their call for proposals, but you might be able to email them and get help. So that would be one way to do it. Or if you have data sets, you can contact us and we can try and work with you on it. So the exobiome team isn't necessarily just focused on the more specific set of applications that JGI is. So, and especially if you have a data set that's maybe 50 terabytes, they'd be interested in working with that just to hit some of their milestones for the project.
Okay, great. We get a couple questions. Now we get questions, and maybe we'll take one more and then we'll defer the rest into the breakout room. One is from Jeffrey Blanchard. Could you comment on the future of contig-based versus MAC-based annotation and mapping?
So I guess right now right now we're doing contigs, then annotation, then mags, and then you tie the annotation back, mostly for the taxonomic information. The problem is always simpler if you can split that out ahead of time. So it's possible it could go there in the future. Really, what would be nice is, unfortunately, right now we're having to do both. So we're doing this multi-assembly approach, and in some cases we're doing the combined assembly approach. So what would be nice, at least short term, if for some of these projects we could kind of get rid of the individual approach and just do a combined assembly approach. It kind of depends what researchers are doing. The thrust in the last couple of years has really been from gene-centric approach to genome-centered approach. And so I think, following that trajectory, ideally you have everything separated out before you go into annotation.
Yeah, thank you, Alicia. So Dylan will take your question in the breakout room. We hope to see you there. And I think we'll move on to the next session. Thank you so much, Alicia, for an excellent talk.
Thanks.
Thanks. All right, let's move on to our next speaker. We now have a set of shorter talks that were selected, as Frederick mentioned, from the abstracts. And I'm going to start welcoming Dariia Vyshenska. She can come on the stage here. Hi. Dariia is a postdoctoral fellow in environmental genomics, and that is within the JGI Metagenome program. Her research is focused on developing a scalable framework for the analysis of DNA stable isotope probing metagenomics data, and that allows culture-independent identification of microbial populations performing specific metabolic functions. And she's going to talk about this super exciting technology and topic today in her talk that's entitled DNA SIP Metagenomics, Linking the Identity of Bacteria with Their Function in Complex Microbial Communities. Dariia, take it away.
Thank you for a great introduction. Let me share my presentation first. I'm also gonna switch off my video, but because I'm experiencing some issues. Okay, I hope you see my presentation.
Looks good.
Okay, perfect. Hello, everyone. Thank you for attending my presentation. I really appreciate that. Today I'm going to be talking, as Tanya said, I'm going to be talking about the new JGI service, DNA SIP Metagenomics. I'm sorry. Okay. One of the main goals of bacterial ecology is linking the identity of microbes to their function. For example, we might be interested to find which microbes in the microbiome decompose specific substrates. But what do we do if we work with uncultivated microorganisms? We cannot culture them. This is where stable isotope probing is useful, in short, SIP. So I'm going to be referring to it as SIP. Let's look first at what is SIP actually. SIP is a technique where we start by dividing our microbiome into two groups, control and treatment. We add a regular substrate of interest to the control group, and we add a labeled substrate to the labeled group. We also can call it treatment group. The substrate can be labeled with stable isotopes of carbon, oxygen, or nitrogen. After we add the substrate, we wait while the bacteria in the microbiome processes it and incorporates heavy isotopes into their nucleic acids. Once that is done, we can go on and extract DNA from our samples. Now, we have two identical microbiomes, but only one group received labeled substrate. So we expect active bacteria in the treated group to have heavier DNA because it contains stable isotopes. Since it's heavier, for active bacteria, we should see a shift in the distribution of the DNA along the density gradient. To detect this shift, we basically spin the DNA for several days and collect fractions for each replicate sample. Once we collected the fractions, once that is done, we can then create libraries for each fraction, sequence them, and analyze the data to find those bacterial incorporators. So how will this service be represented at the JGI? The users will be working with their microbiome and substrate of interest. Once the incubation by our users is complete and DNA extracted, the DNA samples can be shipped to the JGI facility. Here at our facility, samples will be spiked, fractionated, transformed into libraries, sequenced, and data will be analyzed. The user will be able to download the analysis results and all related data files if the user wants to reanalyze them or check or to do anything they like to. Now let's talk about what is so special about our framework. The whole protocol can be roughly separated into two portions: sample prep and data analysis. Rex's team at JGI have developed an automated metagenome-oriented sample prep workflow. It allows identification of mishandled samples by using synthetic labeled pre-fractionation spikings. Additionally, we introduced post-fractionation unlabeled spikings that are used in data normalization. We also developed an automated metagenome-oriented data analysis workflow. We are able to automatically identify mishandled samples, generate reference metagenomes, and work with the absolute bacterial abundances. All those new features improve the reproducibility of DNA SIP experimental results and allow scaling of the method, which is very important for the JGI. Our data processing pipeline looks the following way. We start from quality filtering for the raw reads, then a two-way assembly, a gigantic co-assembly with MetaHipMer, so talking about the question about MetaHipMer, and single fraction assemblies with Metaspades, binning, bin deduplication. We then remove mishandled samples based on labeled spikings data. We map the reads to reference metagenomes to acquire bin coverage, normalize the coverage, and perform QSIP analysis to identify statistically significant bacterial incorporators. So we also tested our framework. To test it, we spiked the freshwater bacterial community from a local pond with two bacterial isolates, Escherichia coli and Pseudomonas putida. We variated the amount of labeled isolate DNA added to unlabeled isolate DNA that we spiked into the samples. And we also variated the ratio of the labeling for the labeled DNA. In total, we created 7 different levels of treatment, with each level having at least 2 replicates. Unfortunately, as I mentioned, we had E. coli and P. putida, but we were unable to detect P. putida reads in a control group, and therefore were unable to perform the analysis for that particular isolate. This was due to technical issues with the samples, but we were very lucky to have the data for E. coli. And so we got the results for this isolate. In conditions where we had 2 replicates, we either had a significant amount of false positives, like the here, or could not identify E. coli as incorporator, like here. But we did identify E. coli as the sole incorporator for the conditions that had 3 or more replicates, and those are 5 conditions out of 7. To summarize, we have developed, scaled, and successfully tested the metagenome DNA-SIP framework that can be instrumental in linking functions to bacterial identity in complex microbial communities. And we now provide it as part of the JGI services. And at the end, I would like to thank my colleagues from Metagenome Science Program, Emily, Simone, Mich, and Maureen, as well as our internal Rex and his team and external collaborators, Ryan Ziltz, Jennifer Patridge teams for amazing work they have done for this project. And of course, JGI and all the staff, especially Alicia for MetaHipMer assembly. And lots of other people who contributed to the project. That's it for me. And if I have some time, I would be happy to answer questions. If not, then
We'll move all for the short talks. We'll move all the questions into the breakout room. So come meet us there, and we'll have a lot of questions there. I'm sure a great talk and exciting technology. Thank you, Dariia.
Thank you. Bye bye.
Bye. We'll move on to the next speaker, which is George Schaible, on the topic of exciting technology. This just keeps going. George, he's pursuing his PhD in biochemistry at the Montana State University as a molecular bioscience fellow under the mentorship of Roland Hatzenpichler. In 2020, he won a Future Investigator in NASA Earth and Space Science and Technology Grant. It's called the FINEST Grant for people that are familiar with this. And that's supporting his graduate research project, studying the biology of multicellular magnetotactic bacteria. And that's what he'll talk about today in his talk that's entitled Correlative Raman-FM-SEM-EDS-nanoSIMS Links Identity, Biochemistry, and Morphology of Environmental Microbes. Take it away, George. Thank you.
Awesome. Thank you. And you can hear me just fine, right?
Yes, everything looks perfect.
Okay. Perfect. Thank you for hosting our talk. As Tanya introduced, I'm going to go over a little bit of how we're applying multiple different methods to study environmental microbes. And this work has been done in collaboration with Anthony Coates, who gave a talk 3 weeks ago, I believe, introducing a new lineage of archaea that he gave to the NeLLi, as well as John Cliff, who's from EMSL at PNNL, and of course, my advisor, Roland. So moving into this talk, basically, I want to frame the question that we're addressing here, and that's looking at the diversity in what are seemingly homogeneous communities, specifically environmental communities, kind of shown here under this magnifying glass. And the issue is that when we look at microbes in the environment, often there is a greater diversity that exists than we can observe with a single method. And so if we're able to look at them using multiple methods, we would see that there's a greater diversity existing in this environmental sample that is hidden when we look at it using only one single method. So the issue is that we need to overcome this limitation by finding an approach that will correlate these different methods to provide a deeper look into the microbes that we study. So the solution is to correlate those modalities into a single sample, allowing us to look deep into what we're observing. And currently there's no single instrument that allows us to do this. So by taking a sample and applying multiple different methods onto that single sample, we're able to study microbes on a single cell level in depth. So to start, I applied this to my project, which is the multicellular magnetotactic bacteria. And I just want to give a slide here introducing these organisms. So I just break down the name here, and they are multicellular in that they're composed of several cells of the same species, as you can see in all these micrographs here. In this SEM, false colored SEM image here, these little lumps are single cells that are living together in this entire consortia. They are also magnetotactic, meaning that they're able to sense Earth's geomagnetic poles, and they do this by synthesizing minerals inside of a bacterial organelle referred to as the magnetosome. And you can see those minerals in this backscatter electron image down here with these little white dots that are in a line. And these essentially act as a compass needle, allowing the organism to sense Earth's geomagnetic poles. My specific organism synthesizes greigite, which is an iron sulfur mineral. Often you'll hear magnetite as being one of those paramagnetic minerals as well. And of course, these are bacteria that belong to the desulfobacteriota. And well, they have, we've known about them for almost 40 years. We have still been unable to culture them. And this lends to our interest in correlating these different microscopic methods. So as far as the MMB go, there's many different morphologies that have been observed across the globe. And some of those are shown here. And when we go to our site and we just look at it using a simple DNA stain, we see what we believe to be just a single morphology, which means that we might have just a single organism living in there. So our question is whether there is a deeper diversity existing in our site than what we actually observe when we look at it with traditional microscopy. So one of the first steps we did was we did full-length 16S RNA sequencing through JGI with a metagenome and several single-cell mini metagenomes where just single aggregates where we did full genome sequencing on. And what we found was that in our site, we actually have 5 unique populations of MMB existing. And so, of course, we designed fish probes, fluorescent in situ hybridization, to label each of those 5 populations, 3 of them shown here. And so we're able to now identify these different populations using the fish probe. And while this is helpful, we now know that there's the different species and we can identify them. We wanted to dig a little deeper. The genome suggested that genetically speaking, they weren't entirely too different, but we wanted to investigate that on a deeper level. And so to do that, we applied Raman microspectroscopy to our samples. And we did this, well, let me introduce Raman first. And Raman's a light-based spectroscopic technique that shows the vibrational modes of molecules. So what it allows you to do is identify molecules in a cell and correlate those to specific peaks in their spectra. So shown here is a spectra from an MMB, and this large peak here on the right corresponds to lipids, and you can see proteins as well as nucleic acids. And then here where I label greigite, this is the magnetosome. So we're able to actually identify the type of mineral within these cells. So that corresponds to this label-free reporter that we're able to identify using Raman microspectroscopy. And then as Dariia just introduced, we're also able to investigate isotopes and substrate labeling by using SIP. So we use deuterium, which is heavy labeled water, to investigate activity of microbes. So here I just have some cartoons showing a molecule with just your normal isotope of hydrogen and then deuterium on it. And what this does is this heavier isotope changes the vibrational mode of this molecule, which then can shift the peak in the Raman spectra, giving us a characteristic peak of activity that we can investigate. And this can also be done with carbon-13 and nitrogen-15, so we can study different uptake of these organisms and as far as what they metabolize. So to show you our workflow, what we do then is we can take our environmental microbes, incubate them with a SIP label that the active microbes in our environment will uptake. We can put those onto a sample slide that has a specific orientation, look at these under the scanning electron microscope, and then collect Raman spectra to show the active organisms within our sample, fish label those so that we know what populations are existing in the sample. And then we can even, if we so choose, do a further step and investigate that with NanoSIMS to identify where the isotopes are being uptake in the community. And then correlate that to a single image that I show here. And so this is a mock community that we use to benchmark our method and shows all the different methods superimposed on each other. So we labeled E. coli, which are shown here in green, and then Methanosarcina shown here in this magenta color. And then the SIP label, the stable isotope that was uptaken in this mock community by active cells is shown here as a white halo around the cells. So the next step was to apply this to our environmental sample, the MMB, the multicellular magnetotactic bacteria. So what you see here is a bright field image that we see when we do our Raman microspectroscopy. And so each cell in this image is assigned a number, and we're able to collect Raman spectroscopy data on these cells, showing us the biochemical makeup of these individual organisms. We're able to then do fish on those on this sample, and as well as scanning electron microscopy. And so all this data can then be together superimposed on a single image, allowing us to identify the specific taxonomy, their biochemical makeup, and their morphology, which is thought to possibly be unique to specific organisms within our environmental site. This data, we are currently working on collecting NanoSIMS for this, so that will be something that we eventually add to this data. Kind of closing, I just did want to give a shout out. We did have a image from our work selected for one of the final journal covers in ISME. So if you get a chance, you're part of the ISME community and you can vote, go ahead and vote for that. And then I'd like to just close by thanking our lab, specifically Dr. John Cliff at PNNL, who helped with the NanoSIMS, and Anthony, who's been a huge contributor through the whole project, and also all of our funders. Thank you.
Great, thank you so much, George. Very exciting work. Let's defer again all questions to the breakout room, and we'll move on to the next speaker, which is Ronald de Vries. Ronald is a group leader at the Westerdijk Fungal Biodiversity Institute. He's also professor at the Utrecht University in the Netherlands, and his research is focused on fungal conversion of biomass into valuable compounds. And for that, he uses a combination of genomics, genetics, biochemistry, and physiology. And in his talk today, he's going to talk about coevolution of genes involved in plant biomass conversion by fungi. Looking forward to your talk.
All right, thank you very much, Tanya, for the introduction. And thank you, so, of course, for giving me the time here. So this came up quite recently because this is a new line of research we were investigating and which seems to link quite nicely to some of your initiatives. So I just want to give a short introduction of what we are interested in doing in this context. Let's get the right screen. So basically, the whole idea we got in this, and this of course is built on huge amounts of experimental data, which we've largely generated in the past with the help of JGI. Which gave us some indications that there are maybe some things happening that direct the evolution of biological systems. So when we look at that and we think about these things, we typically consider that the evolution is driven by the biotope of the species or specific stress situations. And we should of course take into account that when we're talking about this in fungi, that's going at a much slower rate than it typically happens in bacteria. But often what we see in fungi, we see that there are two elements: the enzyme and protein encoding genes evolve in response to the actual need that the organism has. And then the regulatory system also evolved to ensure that the correct implementation of these novel enzymes occurs at the right time, so that the fungus has the optimal situation to cope with whatever situation is present in the biotope. Now, a lot of studies, of course, address plant biomass degradation, but not many look really at co-evolutionary patterns. And we have quite nice indications that this actually occurs. And it's something we are now investigating more deeply. So just for those of you not so familiar with plant biomass conversion, just to sort of put it in a little bit of context. So this is a very simplified model of plant biomass conversion by fungi, where you can imagine that there are plant biomass, which consists largely of polysaccharides, which gets degraded by extracellular enzymes into monomeric components. These get taken up in the cell, usually sensed either extracellularly or intracellularly. They then cause this signaling pathway, which activates transcription factors, which then results in the production of metabolic pathways that convert the compound, but of course also in the induction of extracellular enzymes to keep degrading the polysaccharide. So if you think about this process as such, then you basically have four main components. You have the extracellular enzymes that are degrading the polysaccharide. We have the transporters that are taking up the resulting monomers into the cell. We have the metabolic pathways that convert the monomers into the required components for the cell maintenance and growth, and the transcription factors that regulate the process. So what we would like to look at is co-evolution, both within each of those four aspects, but also between them. And so far, we've mostly been looking at the enzyme families. And one of the things that is quite interesting when you look at the enzyme families involved in plant biomass degradation is that we have multiple enzyme families that act on the same polymeric substrate and are needed together to have efficient degradation. These cocktails, which we of course also use in industry, are typically very complex. and the synergy between the enzymes is also usually very complex. So one of the questions we already asked ourselves several years ago, can we see whether the evolution of these enzyme families actually is coordinated, or is it more a random process? And I just want to give you one very simple example here, which is a very old slide, to be honest, which we've made quite a few years ago, where basically when we didn't have so many genomes yet, we just very simply correlated the number of genes per enzyme family for 22 genomes to see if there were any patterns we would see. And some of the things which came up quite nicely is that we see here, for instance, on this side of the correlation heat map, you basically see 6 families which are all involved in xylem degradation, which all seem to be responding in either going up or down in numbers in the same way across these 22 fungi. What's probably even more impressive, well, we see the celluloses one here, but I'll skip those. The one which really caught our eye was the one over here, where we basically see 7 families related to pectin degradation, which all cluster together. Now, there's one family sitting all the way over here. That was a bit confusing at first, but we later realized that this is the only family which actually the enzymes require an acidic pH for activity, whereas these are mostly neutral families. But the fact that we see 7 families all related to the same substrate, which have a very correlated pattern of evolution across these 22 fungi, gave us the impression that this is not a purely random process, but there is a certain level of co-evolution. So at the moment, we basically then have several questions which we'd like to answer. First of all, of course, what evolutionary patterns do we actually identify across fungi? And we want, of course, go way beyond the 22 fungi we tested at first. We want to look ideally at hundreds, if not thousands, of fungal genomes. Looking at things like expansion and reduction of the number of genes and enzyme families. Looking then at functional diversification. I don't have time for that today, but we have very clear indications of functional diversification that occurs after expansion or duplication of genes in enzyme families. Looking both at the diversification in orthologs between species, but also in paralogs within species. Looking at parallel evolution of function, we have evidence for that as well, certainly in the case of some regulators, that completely unrelated sequences result in functional proteins that have practically the same function as the one in the other species. So we do see parallel evolution patterns. And then basically, of course, the bigger question, the overall question is, do these aspects evolve together? So do we see co-evolution, not just within enzyme families, but for instance, also between extracellular enzymes and metabolic enzymes, or metabolic enzymes and sugar transporters, or regulators with any of those other aspects? So we saw it already within the casea genes, but we would like to look at the co-evolution pattern between all of these. Now, this is, of course, something that requires quite extensive computing power and correlations with the increasing number of genomes. So this is something which certainly would benefit also from the infrastructure at JGI. So that's all I want to mention today, because we're just sort of in the starting point, just mentioning the people on our side which are involved. So there are 2 PhD students, Jiajia and Li, who are in my group together with their co-supervisor Mao Peng, who is my senior bioinformatician. And this is all a collaboration with Professor Berend Snell at Utrecht University, who's an evolutionary biologist. And that's basically the people on our side. So thank you for your attention.
Thank you, Ronald, for the very nice talk. For the sake of time, let's move on to our next speaker, which is Robert Riley from our JGI. Robert is a data scientist in the Genome Assembly and Analysis group at JGI, where he applies emerging genomic technologies to fungi, algae, other unicellular eukaryotes, and where his work is currently focused on terabyte scale metagenomes. So you're going to be hearing more about MetaHipMer in his talk, Increasing Taxonomic Diversity of Genome Bins Using MetaHipMer for Terabyte Scale Assembly. Robert, take it away.
Okay, test 1, 2. Can you hear me?
Yes, perfect.
Hello, good. Okay, thank you for the introduction, Tanya. So I'm going to talk about probing the taxonomic depths of metagenomes through large-scale co-assembly. So just for an outline of my talk, I'll just reiterate a little bit of what Alicia talked about earlier. Metagenome co-assembly versus multi-assembly. What is it and what are the benefits? And you know that the benefits include improved assembly contiguity and binning. And then I'll focus a bit on the case of a peat bog metagenome co-assembly, in which co-assembling the data helped us to recover the full taxonomic diversity in the environment and perhaps rare biosphere candidate phyla. So first, let's just cover metagenome co-assembly versus multi-assembly. I almost don't need to because Alicia covered it so well previously, but suppose you have an environment and you sequence some, n samples of it, and then you have n sets of reads. And the typical approach, for a number of reasons, mostly, compute power being one of the main ones, is to multi-assemble all of these samples separately. So that's what multi-assembly is. It's separately assembling all of these samples in a metagenome environment. Often you'll combine them afterwards and possibly de-replicate. So co-assembly is when you assemble all of the samples in one go. Now, it turns out that co-assembly yields more contiguous metagenome assemblies than multi-assembly. So if you line up the contigs from largest to smallest on the x-axis of this plot and count the cumulative base pairs, the higher up line or steeper slope will indicate a more contiguous assembly, and we see that that is the case for co-assembly versus multi-assembly of the same samples. And what we strive for in metagenomics is more contiguous assemblies. We want fully assembled microbial genomes, eukaryotic chromosomes, viral genomes, etc., as opposed to fragments. So that's a good thing in favor of co-assembly. And co-assembly also substantially improves the binning, the recovery of mags. So, Alicia presented the case of this great redox experiment, GRE, in which a whopping 8 terabytes of metagenome sequence data was generated. And as we went to co-assemble incrementally larger subsets up to 8 terabytes, mostly for scientific reasons, but also as a test of whether MetaHipMer could even assemble that much data, and then bin assess the quality. And we find that with co-assembly of larger and larger subsets, we get more and more high quality and medium quality bins. So that's a good thing in favor of co-assembling more data as well. So just to cover the bin, what I mean by high and medium quality bins, these are the standards here, and we can also just think of these as mags. So going forward in this talk, when I say bins, I'll just mean the high quality and medium quality bins as defined by these standards. Notice also these yellow and turquoise lines here indicate the median such bins you might expect in any environment metagenome project, or specifically in soil metagenome projects, so done at the JGI. So as we increase the amount of metagenome data assembled, even modestly, we get substantially more bins than you might typically expect. So that's all well and good, but what good does it do us? So we get more quality bins in co-assembly, but what can we discover in them? So to try to answer that question, I'll focus on the case of a peatland bog metagenome. So peatland ecosystems are, just a few % of the Earth's land surface, but they sequester 12 to 30%, it's estimated, of soil organic carbon. So they have a very disproportionate influence on greenhouse gases such as CO2 and methane. So this proposal here from Eric Liliskov's group aimed to look at the fungal, bacterial, and archaeal communities, in peatland ecosystems and how they might be influencing gas flux and carbon cycling. So what we did, or what the PI's group did, was sample these microbial communities from 8 peat bog microenvironments at different depths. And what we did at the JGI was sequence these samples and multi-assembled them one at a time, the usual way with spades. And we also co-assembled these samples together with MetaHipMer. So what we see comparing the bins recovered in the multi-assemblies versus the co-assembly, we really just get substantially more bins using co-assembly. So with the multi-assemblies, we get a medium of 3 bins in the multi-assemblies. Sometimes we even get 0 bins. And the sum of the bins that we get across all of the multi-assemblies when we combine them is 28, which is obviously considerably less than 118. So this is, just sort of a good indicator of the improved bin recovery in co-assembly versus multi-assembly of the same samples. When we look at the unique phyla detected in those bins, so we see a total of 12 unique phyla found across all of the multi-assemblies compared to 18 unique phyla in the co-assembly. So the improved binning also means improved recovery of the taxonomic diversity in the environment. And when we go and look at what the specific phyla are in those assemblies, the multi-assemblies compared to the co-assembly, some 9 phyla are unique to the co-assembly, so they are not found in any of the multi-assemblies. So those are a starting point for looking at the rarer, undiscovered things that might be in this environment. They also include, just pointing out that the sulfobacteria mentioned a couple of talks ago in George's talk as something that's unculturable or difficult to culture. And I also want to point out FCPU 426. So we found 2 bins from this candidate phylum. And when we went to look a little closer at it, it turns out that a 2012 16S RNA survey of a sulfur spring found this candidate phylum for the first time and used targeted qPCR to determine that it was present at much less than 1% abundance relative to other members of the community. So part of the rare biosphere. Later on in this Dowd et al. Study that this blurb is quoted from, they used a functional enrichment technique to find low abundance cellulose degrading microbes and amplify them using single cell methods, and again found this candidate phylum. So just to define this term rare biosphere, so it's not mysterious, it just means microbes found at very low abundance in environments, so maybe less than 1% of the total. And if we go and look for this candidate phylum that we found in the peat bog, FCPU 426, and that was found in these previous studies of a sulfur and a hot spring and so forth, there's not very much in NCBI for this. So there's really just 1 genome and 7 nucleotide records. So this kind of indicates that it is a understudied and undercharacterized microbial phylum that might be worth further investigation. So to summarize, co-assembly of large metagenome data sets can give you more contiguous assemblies, more bins, and better resolution of the full taxonomic diversity in your environment than multi-assembly can give you. This is not to completely knock multi-assembly. It has its uses. Often it is only the only practical option due to memory constraints discussed by Alicia in her talk. And also multi-assembly is good for looking at the population variation in a sample, whereas those population variants tend to get buried or erased in large co-assemblies. And finally, large-scale metagenome co-assembly may be a useful tool for probing the rare biosphere. And I'll thank you all for your attention and thank everyone who contributed to this work.
Great, thank you, Robert, for an excellent talk. For the sake of time, we're gonna move over quickly to our next speaker and our last speaker for the day. I'm very excited to introduce this next speaker and our closing speaker for the 2021 NeLLi Symposium, which is Tom Delmont. Tom is a CNRS researcher at Genoscope in France, and he's part of the Terra Oceans Consortium. Since early in his career, he has focused on using metagenomics to explore microbial communities. He's carved out his research niche now, intensively investigating diversity, ecology, evolution of bacteria, archaea, eukaryotes, also giant viruses infecting eukaryotes, and specifically in the surface of the ocean, in the settlement surface, and using various tools in the Omics toolkit for cultivation independent studies. Tom also took part in the development of Envio, which is a widely used and pretty neat bioinformatics platform that's been developed in the Marin lab. So today's talk, he's gonna talk about from Terra Oceans to Plankton genomics at large scale. Tom, take it away.
Yes, thank you very much for the introduction and for the opportunity. So you hear me and you can see the slide?
Yes, everything looks great.
Okay, this is perfect. So I will spend the next 20 minutes to tell you a little bit about Plankton genomics from within the frame, the scope of the Terra Oceans expeditions. And so I work at the Genoscope, which is the National Center for DNA Sequencing in France, which has produced, I think, all of the data for Terra Oceans. So Plankton includes millions of lineages of viruses, archaea, bacteria, and eukaryotes. And an easy definition of Plankton is all of those life forms that are so small they cannot fight against the current. So they are at the surface of the oceans and seas, but they don't decide where to go. And they form complex ecological networks. They have produced about 50% of the oxygen on Earth. And here you have a snapshot of diversity of just a few species of diatoms, which represent a tiny part of this diversity. We have a lot of beautiful images for plankton, and especially as presented here for the eukaryotes, but we do not have a lot of genomes. And this is in part because of cultural limitations. And this is, in my opinion, where Terra Oceans is contributing a lot. So Terra Oceans has been collecting plankton across 143 stations in all the oceans and also the Mediterranean Sea and the Red Sea. So this is about 1,000 deeply sequenced metagenomes focused, depending on the metagenome, on viruses, the bacteria and archaea, and the eukaryotes. So in this world map, you can see the scope of the sampling expeditions, which started in 2009 from France. And the latest expedition was in the Arctic Ocean in green in this slide. In each of those stations numbered here, multiple types of plankton was collected. And the next slide is going to explain to you a bit how it was done. And I think it was very useful for the downstream bioinformatic analysis. They did size fractionation so that some filters will be enriched in viruses, others will be enriched in bacteria, especially the 0.2 to 3 micron size fraction. And multiple larger size fractions will be enriched in the eukaryotes. So at this point, the equivalent of 10,000 human genomes were sequenced within the scope of those 143 stations. And it's about 300 billion metagenomic reads. And I think this is a considerable opportunity to better understand the genomic diversity of especially the eukaryotic plankton, because most of those metagenomes are enriched in eukaryotic signal. But first, we need to solve a complex metagenomic puzzle. The way we do it, and many other people, including in the presentations before me, is by means of genome-resolved metagenomics, which uses two key steps to go from short reads to environmental genomes. The first step is the assembly. You can do a single assembly or a co-assembly. And the second step is a binning step. In my case, I use it within the frame of the NVO platform. So NVO stands for Analysis and Visualization of Omics Data. It's a bioinformatic platform that allows you to do multiple things, including within the frame of genome-resolved metagenomics. So for Terra Oceans, what we've done is to interact with the developers of NVO to upgrade it so that it can also identify eukaryotic genomes. Historically, NVO and other platforms are focused on bacteria and archaea especially. And they do it by focusing on specific single copy core genes that are detected in all the bacterial genomes one time or all the archaeal genome one time, but this is also true of eukaryotes. It's just not the same markers. So if you don't look for those, you might miss beautiful environmental genomes of eukaryotes. So we have included this database of marker genes for the eukaryotes. And now you can recover bacterial, archaeal, and eukaryotic genomes from within NVO. So after this, we applied it to Tara Ocean's metagenomes. We started with 280 billion metagenomic reads. We organized them in 11 oceanic regions. One, for example, would be all the metagenomes of the Mediterranean Sea in blue. And from there, we performed 11 large co-assemblies. So this is perfect because we have been discussing this in the previous talks. I also believe there is a lot of value to the co-assemblies. It's also increasing complexity, so it's not always a good idea. But I think in this particular case, it was beneficial to us. From those 11 co-assemblies, we produced 12 million contigs of sufficient length. And from there, we used NVO to manually bin those assemblies using sequence composition and differential coverage, and then inspect manually each of the environmental genomes, which are called MAGs for metagenome assembled genomes. So we individually inspect all of those MAGs to curate them if needs be. At the end, we created a non-redundant database of about 700 eukaryotic MAGs. Not all of those MAGs are perfect, and it's much more complex as compared to bacteria and archaea today because eukaryotes have larger genomes and more complex genomes. So in this case, for example, the average completion is only 40%. Yet many of those MAGs are more than 90% complete. Some are very low completion. So this is an example of curation of one of those MAGs. If you look at the inner tree, it's a clustering of all the contigs from this MAG, and then the layers represent individual metagenomes from the co-assembly. So you will do individual mapping on those contigs and look at coverage values. If there is detection, then you have a black bar. If there is no detection, it's a gray bar. So you can clearly see that all the contigs are nicely correlating across metagenomes in this particular case, which is supporting the biological relevance of this MAG. And I will show you another example. This is just another MAG from the same co-assembly, this time detected in other metagenomes, but again, with a perfect correlation of contigs across metagenomes. So even though this is now about eukaryotic environmental genomics, we can still see beautiful signal. And in fact, it's working quite well, I think. Now, a few of those MAGs were matching to known genomes from cultivation. And I will show you two examples. One is Ostrococcus lucimarinus. It's a model organism for which we have a culture and a genome that is 13 megabase per long. We have a matching MAG, which is 12 megabase per long, a slightly less, with a completion estimate of 80%. So this is quite coherent. And then when you compare the two genomes, the MAG is aligning to the culture at about 97% of the MAG can align to the culture with average nucleotide identity, INI, of 99.5%. In other words, we have a nearly perfect match between environmental genomics from metagenomes and cultivation. The second example is Micromonas pusilla, for which the culture is 22 megabase per long. The MAG is 19 base per long, sorry, with an estimated completion of 75%. And in this case, even more of the MAG is aligning to the culture with a very high identity level, INI of more than 99%. So every time we had a match between a MAG and the standard genome from cultivation, everything was perfectly making sense. The only difference here is that the MAGs are much more fragmented as compared to the culture. This is the main limitation. Often with cultivation, people resolve chromosomes. And at this point, with just the Illumina sequencing short reads, we do not resolve that. We have fragmentation. I think moving forward, it will be solved with long read, maybe nanopore, maybe PacBio, maybe another technologies that will emerge. But at this point, the MAGs are very fragmented. Now, only very few of the MAGs were matching to known cultures. Most of the MAGs were quite novel, and I will spend 2 minutes to discuss one MAG I think is quite interesting. It's a mag matching to a known species, which is called Odontella westflogi, but for which there was no transcriptome or genome sequence at the time. And this mag was recovered from the Southern Ocean, for which there was a major bloom of the species in those two stations, 87 and 88. And in fact, with Tara Oceans, they do a lot of imaging, and we could connect imaging from this blooming diatom species with the mag, which I find quite good, because usually I've been working for a long time with bacterial mags, for example, and I never got any imaging linking bacterial mag to cells, but now it's possible with the eukaryotes because they're much larger. Anyway, this, in my opinion, is a remarkable mag, and I will try to explain to you why I think so, giving you some perspective. The human genome is about 3 gigabase per long, and in comparison, diatom genomes are in general about 30 megabase per long. Some are a bit smaller, some are a bit longer, but this is more or less the average that we know about from cultivation. This mag of Odontella westflogi was 1.3 gigabase per long. As far as I know, this is the longest mag ever recovered, and it's the first one to reach this symbolic length of 1 gigabase per. It's of course dominated by repeats, and I think there is a fantastic story there, and we are working on it. It's about retrotransposons that have been bursts of retrotransposons that we think are 1 million years old, and there is multiple of them, so it's possible this species has a genome that is growing in length over time. The thing is, Odontella westflogi was cultivated years ago, also from the Southern Ocean, by people completely unrelated to Tara Oceans. We reached out to them, they're in Germany, and we asked for DNA, and they sent us DNA. We deeply sequenced this DNA, and this time we recovered a 2.7 gigabase per long genome from cultivation, even more ratio of repeats, but it was completely confirming everything we saw with the mag. And the average nucleotide identity was 95%. So they're not nearly identical, but extremely similar. And I think both belong to the same species name, whatever the scope of species means. And so we could, in this case, use metagenomics to guide our way into sequencing the genome of a culture. Now, I'm done with this little story of one mag. Going back to the database, we have nearly 700 mags, and we wanted to place them in the tree of life using reference databases. So what we did was to generate a phylogenetic tree by including all the mags we could, but also hundreds of transcriptomes from reference cultures from Plankton. The database is called MetDB for the reference transcriptomes, which is a new version of MMATSP for those of you that know about it. Now, the inner tree, so it's a phylogenetic tree based on RNA polymerase genes. And then the first layer tells you about the main groups. A big group, for example, is the stramionophiles that include the diatoms, and the diatoms include this one very large mag I was telling you about just before. But also, for example, we have a lot of mags corresponding to copepods for the opistoconta at the bottom right of the slide, of the figure. The second layer tells you about the mags or transcriptomes that correspond to phytoplankton, so those organisms that are capable of doing photosynthesis. And this is based on a few gene markers. And the last layer tells you if it's a mag from Tara Ocean in red or a reference from cultivation in gray. And then the inner tree, you can see some clades are in red. Those are to emphasize lineages for which we have only environmental genomes, so those mags without cultivation. So this is as expected. The same we could see with bacteria or archaea. Of course, environmental genomics is expanding our understanding at the level of genomics of the eukaryotes abundant in the sunlit ocean. So this is about the history of those abundant unicellular and for some multicellular eukaryotes abundant in the sunlit ocean, but we also wanted to be a bit closer to ecology, and we wanted to focus more on the functioning of those genomes. We identified a bit more than 10 million genes in those genomes, and we organized them organize the genomes based on their functional profile. So if two genomes share many functions, they will be put together. It has nothing to do with where they come from. It is more about what they are capable of doing today. And this is what we observed. So this is a functional clustering of unicellular eukaryotes abundant in the sunlit ocean. And then at a fine grain, you can see coherence with evolution. For example, all the green algae will be together. So all the micromonas will be next to each other. All the X genera, if you focus on one genus of diatoms, all the mags will be next to each other. But when you look back at the main trends of this tree, you can see the emergence of 4 clusters, which we called A, B, C, D. And for example, if we focus on cluster D, it is connecting 3 distantly related lineages. Ocanthoecida, which is Opisthocantha, Oomycota, which is Traminophiles, and a sister clade to Cryptophyta. So this is Cryptista group. And if we switch to the cluster C, you can see a connection between the diatoms that are Straminophiles and the Archaeplastida. So again, those include all the green algae that are very both are photosynthetic, but they are very distantly related. So now we can see a strong connectivity in terms of functions between diatoms and green algae. And the same goes with group B. So this is based on, again, 10 million genes and many thousand functions. So then we run some statistics to look at what are the functions that are differentially occurring between the groups. And we found thousands of them. The thing is, all of those differentially occurring functions in yellow or light orange cover all the main cog categories. So in fact, it's quite a complex story to understand, and we are very far from understanding this. We just see that there is a broad range of functions that are differentially occurring between those main functional groups. And so we simply stated that the properties of thousands of differentially occurring functions suggest that eukaryotic plankton's complex functional diversity is vastly intertwined within the tree of life as inferred from phylogenes. So to summarize a little bit this story, we performed very large co-assemblies, so up to 38 billion metagenomic reads in a single co-assembly. We recovered the first gigabase scale mag, as far as I know. We characterized more than 10 million genes from all of those genomes. And something I personally think is insightful, we found a functional repertoire convergence of distantly related eukaryotic lineages that are abundant in the sunlit ocean. At this point, we have a preprint, which was put out first into 2020. I want to mention that other research groups are independently also recovering a lot of eukaryotic genomes from Tara Ocean's metagenomes. So I think now is the time where we are mature enough to really go into environmental eukaryotic genomics all together as a community, and I think it's exciting. Yeah, I have 3 more minutes, so I want to very briefly mention a very small extra story with you, if you have the time for it. So when we did the binning to extract the eukaryotic genomes, of course, we also looked for other mags, and specifically, we found a lot of bacterial mags that were for many of them, enriched in the large size fractions. A few archaea, but mostly bacteria. And this led to working a lot on this known genus, which is called Trichodesmium. So Trichodesmium is a photosynthetic genus, and all the known species from cultivation are capable of fixing nitrogen, which is very important. And in fact, Trichodesmium is very well known because it's people think it's the main source of nitrogen in the sunlit ocean. We can see blooms of Trichodesmium even from space, and they are often under the form of filaments and colonies that can nurture their own microbiome, which I find quite interesting. They are known for a long time, and because of the structure of those Trichodesmium colonies and filaments, they are not in the free living size fraction. So until now, people could not get a mag for Trichodesmium because they were focusing on the bacterial size fraction, and now we have applied binning to the large size fraction. So we got lucky, and for the first time, we got 5 genomes of Trichodesmium. 3 of them are perfectly matching to known species from cultivation, Erythraeum and Djibouti. And two of them represent new species that we call Candidatus, Trichodesmium miru, and nobis. And what's interesting and why we dedicated time to it is because those new species appear to be missing all the genes related to nitrogen fixation. Not only this, but they also miss many genes that are also related indirectly to nitrogen fixation, especially hydrogen recycling and a few other things I don't show in this slide. This is very unexpected because Trichodesmium has been studied for a long time, is very well known, and never ever anyone suggested there could be some species that lost the capability to fix nitrogen. So we did a lot of double checking. We have been searching for NIF genes signal by doing just mapping, not binning, and we could not see any. So it was supporting the findings. And we also did targeted genome result metagenomics. So what you do is now you know the distribution of a MAG. Instead of doing a big co-assembly of everything, now you can do just a smaller assembly of only the samples in which this particular genome is detected. Often this can improve the statistics of the genome. So we have done it multiple times. Every time we recover a near complete MAG for miru or nobis, and again, we see the exact same statistics. No signal at all for the NIF genes and other genes related to nitrogen fixation. So from there, we were confident enough to claim that there are non-diazotrophic Trichodesmium species abundant and widespread in the sunlit ocean. In this map, every station in blue is a station in which we only detect non-diazotrophic Trichodesmium species. So in other words, those species are expanding the biogeography of this genus in the sunlit ocean. And to some extent, it has implications in terms of ecology and our understanding of the nutrient cycle and climate. Okay, I will finish by, I have a lot of people to thank because of course, especially the eukaryotic story has been done by many people, a lot at Genoscope, but also beyond. I want just to focus on Damien Hinsinger and Morgan Gaia. They were both postdocs at the time at Genoscope, and they have done a lot of work on this story. I will just briefly thank Olivier Jaillon, I've been working with for a long time, Eric Pelletier and Patrick Winker. I want to thank CNRS, the Tarocean Consortium, and the Genoscope, and I thank you for your time.
Excellent. Thank you, Tom, for a really great talk. We have time for questions. I'm checking the Q&A. Nothing has come in yet. A quick question on the nitrogen fixation genes. Those missing genes, could they be on plasmids, or is that?
Yeah, it's a good question. The answer is no, because we did not see any signal for those genes. Those genes, I don't want to take too long on this. I'm really excited about this story. And it was just accepted in PNS, so this will be out soon. But we really looked for those NIF genes, regardless of is it in a MAG, is it in a small contig, and so on, and we could not see any of this signal. And those NIF genes are very stable over time. We even looked for NIF genes that will not be from Trichodesmium because maybe it's a lateral gen transfer. There is nothing like this. It really looks like it's a loss of many functions related to nitrogen fixation.
Okay, cool. No questions in yet, so I'll just keep going. Let's see, quick question on the size fractionation. So you mentioned the size fractionation. So first of all, you have 700 eukaryotic MAGs, which is just an amazing data set. Are they generally from the larger sized fraction, or so they are the large metagenome fraction? And then the other question would be, how do you do the size fractionation? I presume that's with like freshly sampled material on board of the ship, or how is that being performed? Can you comment?
Yeah, so the size fractionation is totally done on board. Okay, every time. So I've never been part of the expedition, to be honest. So, but I know a little bit how it's done. And sometimes they will do filtration just for one size fraction. And sometimes they will do one after the other after the other, different filters with different hole sizes. So the first one will keep only the very large eukaryotes. The second one will keep smaller eukaryotes and all the way down to the viruses.
Okay.
So then you have a filter with biomass for each of those size fractions. This is stored, sent back to Genoscope for sequencing. So your question about where are the eukaryotes detected, it's a good question. So the copepods are small animals. They're quite big. And those were clearly detected only in the largest size fractions. On the other hand, Micromonas, Bathycoccus, those have they are barely bigger than bacteria. So those are also, they are in between the bacterial size fraction and the smallest of the eukaryotic size fractions. So when people say this is a bacterial size fraction, it's a simplification. It's not true. There are particles of giant viruses. There is, of course, some archaea, and then there is a lot of pico-eukaryotes. So it's a mix. And in the larger size fractions, you have the bacteria that are symbiotic. You have trichodesmium. So it's a mix. And the key part is to be able to solve this metagenomic co-assembly that is a mix of multiple domains, and to independently extract bacterial marks, archaeal marks, eukaryotic marks.
Yeah, great, thank you. We have a couple questions that came in through the Q&A. The first one is from Igor Grigoriev. How fragmented are the eukaryotic marks?
Yeah.
I don't know if you have a contig N50 or something.
Yeah, it will vary a lot. It will vary a lot. Some are very good, never to the level of chromosomes, okay? No. But then some are extremely fragmented. So some will have N50 of 3 kb. Okay, it's not a lot. Even though the minimal length of contigs is barely below that. So some will have an N50 much higher than that. The very big diatom mag I was telling you about, that is more than 1 gigabase pair, I think it's 400,000 contigs.
Wow. Yeah.
Yeah. But still, the funny thing, it still allows you to do a lot, because you can still identify the genes. Yeah. I mean, with fragmentation, you cannot do centeny, but with eukaryotes, there is not a lot of centeny. Things are not in operands the same way they are with bacteria. Of course, it's always better to resolve all of this complexity and to go to the level of chromosomes. But for what we did, phylogeny, biogeography, functions, it's not impacting too much. The limitation is the completion of the max, of course.
Yeah. And then another question relates to fungi. Have you been able to detect a range of fungal mags?
Yeah, it's funny. We have one fungal, a very good quality. We even assembled it from multiple regions. So we had redundancy. We kept only the best one. So we have only one that is really a fungi.
Okay.
So not a lot.
Yeah, we'll take one last question and then I think we'll close out. That's from Anastasia Giodi. You mentioned a large fraction of histocons in the tree you showed. Any idea how much of these come from fungi?
Well, no. So those are the copepods. It's not fungi. Yeah. So well, let me very briefly bring
back the slide.
Let me do it very quickly. Okay. Because I have the answer in the tree. So let me share the screen again, very briefly. Okay, so do you see my mouse?
Yes.
Okay, this little thing here is a fungi. So most are the copepods, those small animals, and we have this little thing that is fungi, I think. Yeah. And the fungi has one that is a mag, and the rest is from the references. So really, yeah, I'm very sorry if you are looking forward to having a lot of fungi genomes from the Sunlit Ocean. We did not get them.
Yeah. Okay, great. Let's close out, and if there are any additional questions to the breakout room, thank you again, Tom, for a really excellent, very exciting.
Well, thank you for the opportunity.
Yeah. Yeah, thanks. Okay, so let's close out. This concludes the speaker agenda of our 2021 NeLLi Symposium. I'd like to thank all the speakers again for their terrific talks. I'd like to thank the organizing team, specifically Frederick and Brooke, for putting this together. Thank all the session chairs. Importantly, I'd like to thank you for joining us. If you have comments, suggestions, including speaker suggestions, if you would like to give a talk yourself in the future, please contact us. We will continue the NeLLi Symposium, and we hope to have it in person again, as we did in the past at some time in the hopefully not so far future here in California. For this symposium, the talks were recorded. They will be posted. So if you missed a talk, you'd still like to watch it, you can.